In order to tell you about our work on gene therapy to treat deafness and blindness, I'd like to introduce Jessica. Jessica has a disorder called Usher syndrome, type 1F. She was born completely deaf, but received a cochlear implant. And with the implant, together with lip reading, she's quite good at communication. She also was born with no intrinsic sense of balance. She doesn't detect which way is up and which way is down. But she's able to stand and walk by seeing the room around her. Jessica is now starting to lose her vision. She's lost most of her rod photoreceptors, and so she doesn't have night vision. And she is beginning to lose cone photoreceptors, starting in the periphery and moving inwards, so she's developed the beginning of a tunnel vision. Over the next 10 or 15 years, Jessica will lose her sight altogether. No lip reading, no seeing the room around her. So we would like to stop that. We know that the gene responsible for Usher syndrome type 1F is PCDH15, encoding a protein called protocadherin 15. And we'd like to know, what does protocadherin 15 actually do in the ear and the eye? So you know that in the inner ear, which subserves the senses of both hearing and balance, there is a ribbon of sensory cells, perhaps 16,000 in the cochlea, the hearing organ, and 5 more patches of sensory cells, perhaps 10,000 in the vestibular part of the inner ear. If we look at just one of these cells in this sensory epithelium, we can see this bundle of cilia that emanates from the top surface of this one cell and gives the cell its name. It's called a hair cell. So these cilia are the mechanically sensitive part of the cell. And if we zoom in to look at just the junction between 2 adjacent cilia, then on the top right, you can see this really fine filament, only about 150 nanometers long, that stretches from the top of one cilium to the side of the next taller one. And if we imagine what would happen with that filament, the tip link, as the hair bundle is moved back and forth, you can see in the lower left, as the bundle would be moved to the right, these tip links would tend to be stretched. We know now that these tip links connect to mechanically sensitive ion channels at their lower end, so that when tension is increased in the tip link, it simply pulls the ion channel open. Current in the form of potassium ions enters the cell, changing the voltage inside. And this is the fundamental conversion from a sound wave, a mechanical stimulus, to a neural signal. Critically, the tip link is made in part of protocadherin 15. So Jessica has never made protocadherin 15. She can never pull on the channels in either the hearing or the balance organ. We know less about the function of protocadherin 15 in the eye. We know that it's made by the photoreceptor cells in the back of the retina. And in the bottom, you can see that photoreceptor cells, both the rods and the cones, are characterized by having an inner segment at the bottom with metabolic function and an outer segment that contains the phototransduction apparatus. In between, emanating from the inner segment, are these fine finger-like processes that seem to cradle the outer segment. And that's where the protocadherin 15 is. So somehow, the protocadherin 15 is critical for the long-term maintenance of the photoreceptor cell. So what can we do to try to restore vision? Well, the problem with this particular disease is that we can't use a simple AAV delivery of the normal coding sequence, because it's a long protein and the coding sequence is simply too big for a single AAV capsid. But after decades of work, we know a great deal about the structure and function of protocadherin 15. You can see the X-ray crystal structure on the left, and you can see that there are these sort of repeated domains, about 8 of what are called EC repeats, that are sort of like links in a chain. The ones on the left are connected to one essential protein, those on the right to another essential protein, but we wondered whether we might dispense with some in the middle and preserve function. So the idea would be to delete up to half of the EC domains or EC repeats to make a functional protein that can be encoded in a single AAV. Now, this is not as straightforward as you might imagine, because the junction between adjacent EC repeats is highly stereotyped, involving a number of acidic, negatively charged amino acid side chains, like aspartate and glutamate, and 3 canonical calcium ions that are bound to those negative charges and serve to convey force from one EC repeat to the next. But because we know so much about the structure, we were able to design 8 different versions of these mini protocaterin 15s, and then we can test them to see whether any would rescue. Now, another problem is that most AAV capsids won't transduce hair cells, especially not in the primate. But working with Casey McGuire at Mass General, we've identified 2 new capsids that are quite good at transducing hair cells in the mouse and even better in the monkey. So how far along are we? As I mentioned, the X-ray crystal structure has been solved. We've designed and constructed different mini protocaterin 15s, and one of these works to restore hearing quite well in the mouse model. So on the right, you can see the characterization of hearing. The blue curve shows normal hearing. At most frequencies, it takes a sound of only about 30 or 40 decibels to get a response. Our deaf mouse model in black is completely deaf. Sounds of 80 decibels or higher evoke no response. But the deaf mouse model treated with a mini protocaterin 15 has its hearing restored almost to the normal levels. In other experiments, we've shown that we can rescue the vestibular function that's also characteristic of this mouse model. Now, a problem is that the mouse model is not great for the vision deficit. Perhaps the mouse doesn't show much of a phenotype, perhaps it doesn't live long enough. But we've identified another model, a zebrafish, which will be a good model for testing rescue of the retinal degeneration. Zebrafish develop quickly within 8 days of fertilization. They have a fully functional visual system, and they can respond to patterns of light. And we can see, importantly, that within 7 days, the photoreceptors are degenerating. So we have a phenotype and we have a degeneration that we can hope to rescue in a matter of weeks rather than years. So what's the commercial opportunity? Usher 1F is a relatively rare disease. There are about 150 kids born with this each year in the United States and Europe. But the retinal degeneration takes a long time, 20, 30, even 40 years, so that there's a large cohort of patients who have slowly developing degenerating retinas that could be treated. And so this may give us a cohort of perhaps 5,000 that would be treatable. And so we're going to focus on treating the retinal disease. We hope to halt or perhaps even reverse the degeneration. And unlike the cochlear implant that can be a partial substitute for hearing, there's really nothing that can be a partial substitute for the loss of vision. So this is really the only strategy that we know of that could prevent the blindness. The general strategy, I think, is transferable to other disorders. There are 9 different other genes for Usher syndrome, and most of them also have coding sequences that are too large to fit in a single AAV. So we could move on to other Usher syndromes if this works well. At the moment, we filed for IP both on the new vectors and for the mini protocaterin 15 versions. The next steps will be showing that we can prevent or reverse the vision loss in the zebrafish model. We want to see if these get into human photoreceptors, and we'll use human retinal organoids to see if the protein is expressed, localized properly, and causes no toxicity. And then, of course, we'll do studies in non-human primate retina, again for localization and lack of toxicity. And we hope that will lead to a pre-IND meeting with the FDA in the not too distant future. I'll close just by mentioning the team that we've assembled. This includes ophthalmologists, people with expertise in hearing, X-ray crystallographers, AAV specialists, and together I think we've got a pretty good group that can tackle this. So thank you.